Welcome to our lecture on cell and molecular biology. Today, we're going to explore a fascinating topic, the replication of telomeres. We will uncover how our cells solve a fundamental problem during DNA copying and why this process is the ultimate biological clock that dictates human aging and the development of cancer. By the end of this session, you will understand both the biology and the mathematics behind cellular lifespan. To begin our journey, I want you to think about the little plastic tips at the ends of your shoelaces, known as aglets. Their sole purpose is to stop the shoelace from fraying. Our DNA is structured very much like a long shoelace. Every time a cell divides, the biological copying machine cannot quite reach the very end of the DNA strand. If we did not have a protective cap, our actual vital genes would get chopped off with every single cell division, leading to catastrophic damage. Nature solved this by creating telomeres, which act exactly like those plastic aglets. Now that we understand the need for protection, let us look at why DNA shortens in the first place. This is known as the end replication problem. Because DNA polymerases can only synthesize new DNA in the 5' to 3' direction, they absolutely require an existing 3' hydroxyl group from an RNA primer to start. Once the final RNA primer is removed from the newly synthesized lagging strand, the polymerase cannot replicate that extreme 5' end. This fundamental limitation means that with every replication cycle, the chromosome physically loses a small piece of its DNA. Nature solved this massive problem with a specialized structure and a unique enzyme. Telomeres are highly repetitive, non-coding DNA sequences located at the extreme physical ends of our chromosomes. In humans, this is a repeating 6-nucleotide motif, T, T, A, G, G, G. To counteract the continuous shortening from the end replication problem, certain cells utilize an enzyme called telomerase. Telomerase is a unique ribonucleoprotein, meaning it contains both its own built-in RNA template and a catalytic subunit. It binds to the 3' overhang of the chromosome and adds these repetitive sequences back onto the DNA, providing a new platform for replication. Let us visualize exactly how this enzyme works. On screen, you're looking at a schematic of the telomerase enzyme binding to the end of a DNA molecule. Notice the linear, double-stranded DNA on the left, which ends in a single-stranded 3' overhang. The large oval represents the telomerase enzyme. Inside it, you can see the built-in RNA template, labeled TERK, which perfectly matches the DNA sequence. The enzyme uses this template to add new TTAGGG repeats to the 3' end, shown by the arrow on the right. This extension creates the necessary space for the DNA polymerase to finish its job. This mechanism directly connects to how we age. In the vast majority of normal human somatic cells, the telomerase enzyme is actually turned off. This means that telomeres physically shorten with every single cell division. Once the telomeres shorten to a critical, dangerously low length, the cell receives a powerful signal to permanently stop dividing. This state of retirement is known as cellular senescence, or the Hayflick limit. This shortening acts as a protective biological clock. It prevents old, potentially mutated cells from continuing to divide and causing harm to the organism. While shortening protects us from old, mutated cells, what happens when this clock is broken? In about 90% of all human cancers, Genetic mutations cause the telomerase enzyme to become highly active once again. This is a dangerous reactivation. It allows cancer cells to constantly rebuild their telomeres after every division. Because their telomeres never reach that critical short length, these cancer cells completely bypass the Hayflick limit. They become biologically immortal, capable of dividing endlessly to form massive, life-threatening tumors. Understanding this is key to modern cancer research. We can actually model this biological clock mathematically to predict exactly when a cell will stop dividing. The formula on screen represents this linear shortening. L sub n is the final telomere length after a certain number of divisions. L sub 0 is the initial, starting telomere length. The variable n represents the total number of cell divisions the cell has undergone. Finally, delta L represents the average number of base pairs lost during a single division. Conceptually, the current length is simply the starting length minus the total DNA lost. If the loss rate, delta L, increases, the telomere length decreases much faster, causing the cell to age sooner. Let us see this formula in action with a concrete example. Imagine a normal human skin cell starts with an initial telomere length of 10,000 base pairs. It loses an average of 100 base pairs per division. It will permanently stop dividing when it hits a critical threshold of 4,000 base pairs. We want to find n, the number of divisions. We substitute our known values into the equation. 4,000 equals 10,000 minus n times 100. We rearrange the equation to isolate the term with n, giving us n times 100 equals 6,000. Dividing 6,000 by 100 gives us exactly 60. This means the cell can safely divide 60 times before retiring. Now, how do we mathematically model a cancer cell that is fighting back against this shortening? We must define a new variable, the net loss per division. The formula is delta L net equals delta L loss minus delta L gain. Delta L loss is the natural DNA lost from the end replication problem. Delta L gain is the number of base pairs artificially added back by the reactivated telomerase enzyme. Conceptually, this equation finds the balance point between two opposing forces. If the telomerase is highly active, the gain increases, which drives the net loss down. If net loss reaches 0, the telomere stops shortening entirely. Let us apply our new net change concept to a rapidly dividing cancer cell. We observe a cell that starts with 5,000 base pairs and maintains exactly 5,000 base pairs even after 100 divisions. The natural loss rate is 150 base pairs per division. We want to find the gain rate. We substitute our values into the modified equation. 5,000 equals 5,000 minus 100 times the quantity 150 minus the gain. Subtracting 5,000 from both sides leaves 0 on the left. Dividing by negative 100 removes the multiplier. We are left with 0 equals 150 minus the gain. Moving the gain to the other side, we find the telomerase adds exactly 150 base pairs per division. The length remains perfectly constant. The example we just solved actually reveals a profound mathematical truth about cancer, which we will now prove formally. What is the exact mathematical condition for biological immortality? We start with our net shortening equation. For a cell to be truly immortal and bypass the Hayflick limit, its telomere length must remain perfectly constant over time, no matter how many divisions occur. Mathematically, this means the final length, L sub n, must exactly equal the initial length, L sub 0. By substituting L sub 0 into the left side of our equation, we set up the foundation to find the exact balance point required for endless replication. Continuing our derivation, we can now find the exact condition for immortality. We subtract L sub 0 from both sides, leaving 0 equals negative n times delta L net. Because the cell is actively dividing, the number of divisions, n, is greater than 0. Therefore, the only way this equation holds true is if the net change itself is exactly 0. Expanding the net change term, we get delta L loss minus delta L gain equals 0. Moving the gain to the other side, we arrive at our final proven result. The gain must exactly equal the loss. This proves that cancer cells achieve immortality by perfectly balancing natural DNA loss with telomerase-mediated DNA addition. We have covered a lot of ground today, so let us summarize the key takeaways. First, the unavoidable end replication problem causes our linear chromosomes to lose DNA with every division. Second, the telomerase enzyme provides a brilliant evolutionary solution by extending our protective telomere caps. Third, while normal somatic cells use this shortening as a programmed biological clock to prevent mutated growth, cancer cells maliciously hijack the system. By perfectly balancing DNA loss with telomeric activity, they achieve biological immortality. Understanding this delicate mathematical balance is absolutely crucial for developing future therapies.